And then also for questions, like anybody on the team can answer them, right?
We hope that everyone participates.
Yeah, okay, cool.
Okay, so go whenever you're ready.
Okay. Good afternoon. My name is Robin. I'll be the first speaker on behalf of our gemstone team, Team Virus. Also speaking today, we'll have Flo and Paul. And Virus, our team name, stands for Viral Investigation in Rat Cells to Understand the Lymphatic System. I know, it's a little bit of a stretch. A little preview of our presentation. We're going to split it up into 5 W's. What, who, why, when, and where. So I'll be starting us off with a little bit of what the overarching scope of our project is, who was impacted by what we're looking at. Then I'm going to pass it on to Flo. He's going to tell you guys a little bit about why we're doing what we're doing, our hypothesis and aims, and some expected results. Paul will finish up the presentation with some closing remarks and a quick summary, and a little bit of like logistics of how gemstone's going to work for us. Our overarching idea is on Alzheimer's disease. Now, we're not trying to cure Alzheimer's disease, but we do know that it is a problem. Alzheimer's disease is a progressive form of dementia. It can impact degradation of your memory, personality, and it can also decrease your cognitive function. Now, the increase in risk of Alzheimer's disease could be due to increased plaque buildup in the brain, which could not have been removed entirely, which could then lead to misconnections, and then you don't make the same connections as before, which leads to your decrease in cognitive function. There are a few risk factors that do impact this, including your age, gender, some genetics, maybe your wealth, and that's not all that it's limited to. So Alzheimer's disease is on the rise. In 2010, 4.7 million Americans over the age of 65 were infected with the disease. This is a graph that kind of projects how it might increase by the year 2050, when 13.8 million people could be diagnosed with Alzheimer's. So 4.7 million to 13.8 million, that's a lot, and we kind of want to say, all right, maybe there's a way that we can help prevent this number from increasing. So besides the people who are actually infected with Alzheimer's disease, there are a few other people who are impacted, including careholders, family, friends. But this is a serious issue. Every 67 seconds, somebody develops Alzheimer's disease. Since I've been talking, at least 2 people have been diagnosed. The number of deaths since 2000 have increased 71%. 71%. Have any of you guys, do you guys know any family members or friends who have been either infected or have passed away because of Alzheimer's disease? Yep, I have 2. Actually, I lost 2 great aunts in the past year to it. So it's an issue. 1 in 3 seniors die from Alzheimer's disease or another dementia-related disease. Now, this is obviously going to cost money, as most medical situations do. It costs about $47,000 a person for care, and if you need any medical assistance with this, which creates a current overall total of $226 billion for everybody infected in the U.S. This number is projected to increase to $1.1 trillion by 2050. Now, there might be a reason why Alzheimer's disease is on the rise, and it could be due to a malfunctioning of the lymphatic system. The lymphatic system is kind of new, and it's a waste removal system in the brain. The way it works is the glial cells or astrocytes, which kind of comprise the system, they extend onto end feet, and on the end feet you have your aquaporins. And in the aquaporins, their job is to kind of facilitate the diffusion of cerebrospinal fluid throughout the brain, which removes the previously mentioned beta amyloid plaques and other plaques that could build up in the brain. And when this system maybe doesn't work too well, Alzheimer's disease could increase the risk of getting it. This is a diagram to help you kind of better understand how it works. So the glial cells are the orange ones. You can see they kind of extend out and have a little flat edge, which are their end feet. Aquaporins are found on the end feet, and we're going to look at the M23 tetramer protein on the aquaporins. So the aquaporins kind of facilitate this flow of fluid, which removes all the plaques from the brain. So there's actually a correlation with herpes simplex virus type 1, which is also known as oral herpes or HSV1, and infection of that leading to an increased risk of Alzheimer's disease. A little background on herpes type 1, it's a neurovirulent enveloped double-stranded DNA virus. And it actually has been shown that it causes a downregulation in aquaporins, which again facilitated the diffusion of cerebrospinal fluid throughout the brain. So we're kind of looking at how the HSV infection on a little cell model of the glial cells, how that'll impact specific protein to see if that might be the correlation with the downregulation of aquaporins. So I'm going to pass this on to Flo. He's going to tell us a little bit about why we're doing it and how we're going to go about doing it.
Okay, my name is Florian Rafade. Now, this is the why of our experiment. This is the essential purpose of why we're doing this experiment. So, first of all, the purpose of this experiment is to essentially analyze the connection between HSV and Alzheimer's disease. There is a gap in knowledge between HSV-1 infection and Alzheimer's disease. And this is very important because HSV has been shown to lead to a downregulation of aquaporin-4 within the lymphatic system. Now, there is no explanation of this downregulation. I repeat, there is no explanation of this downregulation. And this is what we want to study. We want to study the M23 tetramer because we believe that this leads to the downregulation. The M23 tetramer essentially coagulates the aquaporin-4 within the lymphatic system, leading to a swipe of cerebrospinal fluid throughout the lymphatic system, essentially clearing the lymphatic system of amyloid beta proteins. However, we believe that this tetramer essentially is the cause of this downregulation, and this is what we want to study. We want to study the mechanism behind M23 leading to the downregulation. Now, our hypothesis is the M23 tetramer of AQP4 within HSV-1 infected astrocytes will experience a reduction in production or a change in quaternary structure which could contribute to the malfunction of AQP4. Now, this leads to our three aims. These three aims are consequential of each other. So our first aim is to essentially locate the M23 tetramer protein, followed by studying the effect of HSV-1 infection on the M23 tetramer. Our second aim is then to analyze the structure of the M23 tetramer, specifically the protein folding capability of the M23 tetramer. Also, we want to study the RNA structure too. We want to analyze the different amino acid sequences within the AQP4. And this leads to our third aim, because then from this we want to use computer programs to model the mutated M23 tetramer, or to analyze the new change in amino acid sequence within the aquaporin-4. So our approaches to aim one, first to infect the cell culture, would be to use immunoperoxidase staining. This is essentially allowing antigens to bind to different CPE and monolayers within our cell culture. This will essentially allow us to detect HSV infection within our culture, knowing that our culture has been infected with HSV. The next part of our aim one is then to use immunofluorescence staining to have antigens bind to our M23 tetramer to know that there is M23 tetramers located within our aquaporin-4. After that, we will then use flow cytometry, which generates a certain percentage of M23 produced, because it accounts for the new M23 produced and takes a ratio compared to the original amount of M23 in our cell culture. And then lastly, we will then use Western blotting to isolate the different amino acid components of our aquaporin-4, and then we will analyze them. And this will all lead to aim 2. Then in aim 2, our aim 2 is then to examine the RNA structure and then the M23 protein structure. To do this, we will first isolate the M23 tetramer. We will isolate this tetramer by using the blue native gel electrophoresis. This is a 3-pronged system that uses 3 different types of gels: a 3.2% agarose gel, a 4% agarose gel, and a 15% agarose gel. It will allow us to isolate multi-complex proteins in 3 different types of gel solutions, and this will allow us to isolate our tetramer, which we would then use for our third phase. After that, then we will use a PCR in order to isolate the different the different amino acid components of our AQP4, and these both will then lead to aim 3. But for now, with this picture right here, this essentially shows how we will isolate the M23 tetramer. With this BN page, it's an example of how our gels would be arranged. Our gel would essentially be arranged from 3.2% to 4% to 15% to account for the size of the different proteins within our aquaporin-4. And then the the SCS page would then arrange them in terms of a kilodalton scale. So normally a M23 tetramer protein will show up around a 40 kilodalton scale, where it would appear right here and it would be easy to extract and analyze. And then lastly, our third approach is then to use computer modeling programs in order to model the new mutated M23 tetramer and to analyze the new amino acid sequence of our RNA. And we would do this by using HHPRED and HHSearch, which takes an original template that we already know of an amino acid sequence, and then it configures the sequence in a new array to model a M23 tetramer protein which we feel will be mutated, and then it will also generate a new amino acid sequence which we can see. And then from these three aims, for aim 1, our expected result is to see a decrease in production of M23 and a decrease in production of the different amino acid sequences within the RNA. For our aim 2, we would then analyze the protein folding model and we'll see a change in the protein folding model of the M23 and also the RNA level. And then lastly, with aim 3, we will determine whether there is a change with the computer modeling programs, and we can see if there is actually a mutated M23 tetramer or if there's no change at all. And for the next part, I will pass it on to Paul Butts, who will explain more.
Hi everybody, my name is Paul Butts, and I'll be explaining a little bit about the remaining process of our Gemstone team. So we're following the basic timeline for the Gemstone program as regards to the next 2 years. So in this coming spring, following this proposal, we hope to obtain the necessary funds for the remainder of our experiments. We hope to acquire the right amount of resources and set up the preliminary tests for our experiments. In the summer and fall of 2016, this year, we will be conducting our experimentation with the hopes of having enough data to be presented at junior colloquia. The following spring, we will conclude our data collection and prepare for the summer of 2017, where we will begin our senior thesis writing. Throughout the summer of 2017 and the fall of 2017, we will be doing the writing of our senior thesis with the hopes of having it ready to present in the spring of 2018. Our team will be utilizing the laboratory of Dr. Zhang, our mentor, who has graciously allowed us access to it for this project. On the board here, you can see a number of different companies and resources and organizations where we hope to acquire some of our materials and resources. Some of these are open source, such as the computer program which we'll be using, and others cost a large amount of money. With this in mind, we estimate conservatively that our budget will be around $10,000, although this varies depending on when we buy materials, who we buy them from, and the availability. With the closing remarks, we hope to study the effect of HSV1 on the aquaporin 4. In specific, we hope to look at the M23 tetramer and how it is affected by HSV1 infection. We expect that there will be a reduction in production of the M23 tetramer or a change in its structure. Now, with this project, we do not hope to cure Alzheimer's disease, as far beyond the scope of this project. However, this information that we hope to gain can be extremely crucial in developing new treatments and a better understanding of the disease as it is. Future directions for our research potentially include conducting the same research that we're doing now on a live rat model, which we are unable to do due to time and monetary constraints. Now I would like to open this presentation up for questions, and I'd also like to thank Kelsey and Dr. Zhang for their help with our proposal.
All right, nice presentation, nice, concise and clear summary of what you're embarking upon. As our tradition, we let our two outside experts open the questioning. So you guys get to fight it out who gets to go first.
Doesn't matter to me. So you know, I have to say I'm very impressed by what you presented, how much effort you put into it, and I had a chance to read your document as well. So you put in a huge amount of effort for somebody at your stage. I'm actually very impressed. So there are a few things that come to mind. So one is that the rationale for the whole project is based on Alzheimer's and the possibility that herpes simplex may actually play a role. So you may want to look at that association to see how strong you really think that association is. As far as I know, I think that is not widely accepted as a cause of Alzheimer's. Herpes simplex infection of the brain is usually a very acute encephalitis. And it usually affects the temporal lobes. There is a condition that can occur chronically after herpes simplex encephalitis, and that's been recognized over the last few years. It's called NMDA receptor antibodies can develop. And so that can present with cognitive difficulty, which can be chronic condition, and that wasn't recognized till recently. So it's quite possible that people may have mistaken that as being cognitive abnormalities associated with herpes simplex. But that's a treatable condition. You actually plasma freeze these patients and they get better. Okay, so you can analyze them for antibodies to this particular receptor that's present on neurons. You can take that away. So, but that doesn't mean that this is not where I think, even if the association is weak, there are many different reasons as to why to study herpes in the brain, and herpes encephalitis alone may be a good enough reason to study it, right? The other thing is that downregulation of aquaporin as a cause of Alzheimer's, that also you may want to think about, because, so there's a condition called neuromyelitis optica, and so this, it used to be called Devic syndrome, but it's a form in which there are antibodies generated against aquaporin 4. So there are different types of aquaporins, and one of them is aquaporin 4, which is the largest type of aquaporin present on astrocytes. Okay, and so these patients have antibodies to it, and their aquaporin 4 is actually extremely downregulated. You can't hardly find anything. But what they develop, they don't also develop Alzheimer's. What they develop is blindness and paralysis by affecting the spinal cord. So it's a very different clinical presentation. So that's the thing to think about the rationale. So you may say that, okay, well, you know, at least at the beginning, you'll have to be able to say that there are alternative explanations for this disease, and there are alternative pathways that may be occurring, and maybe there is a potential role, but it probably has a bigger role in herpes encephalitis, would be my guess as to what you think about it. The other thing is the experimental design, I think, is good. You've got the virus, you're going to put it on astrocytes. These are, what types of astrocytes are you going to use?
We're going to be using E18 neonatal rats, rat astrocytes.
Rat astrocytes.
Yes.
So the virus is a human virus, right? Human herpes simplex virus?
Yes.
Yeah. So do you know if it infects rat cells?
I believe that it's been shown to affect rat cells. There's been models in the past that have used similar approaches, as far as I've seen.
I'm gonna jump in for a second. If you all wanna sit and help take notes so you all get all this information recorded.
Yeah, I'm taking notes.
You getting all of it? Okay, multiple tappings might be more comprehensive. So just feel free to make sure you capture all of this. Okay, go ahead, sorry.
So, so I'm not a herpes virologist, but one of the things you may want to consider is that you can actually get human astrocytes. They're really available. So that's another thing to consider. You may want to look at the literature and see if there is a difference in aquaporin expression in rats versus humans. The second is its ability to infect rat versus humans, which would be a better model system. You can grow one type of astrocyte, you can grow another one, and human astrocytes are available commercially or through collaborative efforts. Our own lab, we have lots of human astrocytes. We can sign an MT and actually provide you astrocytes to culture if you want. That's not a problem. So that's something to think about. Yeah. And then I wasn't entirely clear on how you were going to look at this M13 is it, or was the
M23.
M23, sorry, M23. So how you were going to model this thing? You expect that there's going to be a mutation in it, or what's going to happen to the M23? Why did you think that it's going to change configuration?
Well, we specifically think that the M23, like, it's been shown to lead to a, well, HSV has been shown to lead to a downregulation of aquaporin 4, but we have been looking that there is a specific ratio of M23 to M1 that essentially keeps the aquaporin attached to the lymphatic system, and when this ratio is changed, it potentially leads to a downregulation. So we want to study whether there is a mutation in the M23 at all, which would possibly lead to a change in the ratio.
So what would produce the mutation?
What would produce the mutation? We felt that the HSV would produce the
Oh, the
Mutation.
HSV
Yeah,
will
by
produce a
infection.
mutation
By
in the chromosome, huh?
yes. The HSV,
How would that happen? HSV resides outside the chromosomal DNA, it doesn't get integrated, right?
but the HSV attacks double-stranded DNA, which we would feel that it would lead to a genetic change within the M23 tetramer.
It is. What, sorry, what were you
Uh,
saying?
it's-
So is there precedence for showing that herpes viruses cause mutation in chromosomal DNA?
I'm not
Do you
sure.
know any
I would have to
literature?
look deeper into that.
I don't know of any precedents for that. I think this was a bit of, I had the same issue with calling this mutational. I think you might be looking for structural changes in the tetramer because, you have a decrease in one subunit and not the other or something like that, but I doubt very much you're gonna see anything mutational in this kind. A mutation means a change in the nucleic acid sequence. I would doubt very much that you'd see anything like that.
Yeah, so you may want to think about that last part of it because it's unlikely you're going to find a mutation. So that doesn't mean that conformational changes may not occur. But I'm not too sure you'll be able to sort that out simply, Even if you just show that the ratio of the proteins have changed and the production has changed, I think that's probably good enough. Do some Western blots and show the change, I think that might be good enough without doing the computer modeling part of it. Unless that's an important part of
Oh,
what
no,
you
it's
guys are planning on doing.
It's a consequential part of AIM too,
Yep.
but it can easily be omitted if necessary.
You have access to computer modeling?
It's the whole thing was what's called open source, so anyone can use it, and it's free. It's just like everyone collaborates on it. It's kind of like a project everyone works on together on the internet.
So who's your computer modeling expert here? All right?
Computer science major.
Oh, okay. You needed one of those.
So that doesn't mean that you shouldn't model it. I think you can still model it and show, okay, these are proteins, this is what they look like, and.
This, we understood like going into this that AIM3 was very dependent on AIM2 and what results we got from that, right?
Yeah.
So, for all we know, we may or may not have positive or negative results, right? If there's no change detected, then that means AIM3 we probably wouldn't go to that. But if we do detect something, then as kind of like an additional step, we put in AIM3 to kind of like ensure, like, is this like a change? Like, can we quantify this? Can we like see what is different and perhaps maybe?
I mean, another thing you could think about is, are there polymorphisms in that gene that are known in the human population?
Oh, you started that splicing and multiple.
Oh,
Yeah.
one of the ideas that we had was the M23 tetramer and the M1 tetramer vary because during gene, what was it, expression?
Yeah.
Sorry, I'm a little rusty on the pronounce there. But the M23 RNA is spliced alternately, as compared to the M1, and so there is a higher percentage or a higher chance of error in that case, in which case we may be able to find something in analyzing that, which could lead to maybe a change in the sequence somehow.
So virus infection maybe affect at the level possibly.
Mm-hmm. Yeah.
We don't know.
Something.
So the reason I bring up polymorphisms is because you could very quickly just search the database, PubMed database, and see if people have looked for polymorphisms. If they have, then you can do all your modeling based on that and say that, okay, and you can hypothesize that certain people who have such kind of polymorphisms are going to be more likely to have less interactions with aquaporin. You could model one protein against the other and see what kind of interactions they may occur and come up with some kind of hypotheses. then it's not entirely dependent upon the other two aims. But if that's an important technique you want to learn, then you could still rationalize it.
So
Makes
kind of
sense.
like in parallel more.
Yeah,
than-
yeah,
Okay.
Right?
Sounds, thank you.
Want to pass on for now?
Yeah.
Oh, okay.
All right.
Okay, and I wanna join you in congratulating all of you for this effort and research. Becoming a scientist is a long process, and this is a great start, I think, to that process for all of you. I had a lot of the same issues that I've written down in this. I have written down most of the stuff that I had. I didn't write down every little thing, but there's lots of writing on lots of the pages here, so I'll give this to you when I
Thank
when I
you.
go.
Thank
You're
you.
welcome to take a look at it and see what you think. But the first issue, I think, was I would not overplay this causative effect here at all. And what was mostly here was a well-written introduction that was mostly hearsay, what I would call hearsay, in the sense that there was not really any strong proof. There wasn't any statistical analysis of something like, for example, here are people with HIV that we have identified as having a latent HIV infection, and those people have a 30% greater chance of getting Alzheimer's. Those statistics would not be very difficult to get. It's very easy to identify people that have a reoccurring latency in herpes, and it's very easy to determine whether those people got Alzheimer's or not later on in their life. So I don't think it would be a difficult statistic to get. The fact that I couldn't find that statistical correlation anywhere.
I think that data is so weak that it's
In
not
the
gonna help too
literature.
much.
What I did find is the same thing that you guys found, which is one paper by one individual who put all this stuff together and is pushing this idea, but I don't think it's well established. I don't want to say it's wrong, but and I'm not a neurobiologist, so I wouldn't say that at all. But this sounds to me like there's got to be a lot more credible people joining on to this hypothesis if it's ever going to move anywhere, right? And the other fact is that herpes encephalitis is quite rare. Alzheimer's is extremely common, right? So, to try to suggest that herpes encephalitis in some way is causing Alzheimer's, it's possible. But if it were happening, it was certainly causing a very small number of those cases, right? Because it's very hard, I think, as far as I know, is that true, to get herpes encephalitis? I mean, it's extremely rare. So be careful about overplaying that correlation. You don't have to, because what you want to do is you're studying an important phenomena anyway, right? You're studying an important idea of what is herpes doing to cells when it infects it, and how is it altering this potentially very important protein in the cells, right? So that's a strong enough, I think, or a strong enough reason to study what you're studying, rather than making. I would put this kind of more as a side note possibility than as a main focus of what you're doing. So I wanted to mention that again, and yeah, so that's pretty much already mentioned. And again, there are several little notes here all on the way that you can take a look at. As far as herpes latency, I think you want to look a little bit better exactly what that is with HSV1. Lots of people get infected with HSV1, almost everybody. Not everybody gets latent reoccurring infections from it, right? So I mean, you have statistics in here that suggest 80% of people have herpes latency. As far as I know, that's actually a lot more people than really do. That's about how many people have gotten infected by herpes viruses. If you look for antibodies in people for herpes virus, we're probably almost all seropositive in the room. But only the few unfortunate ones like me get cold sores all the time, right? I mean, so I don't think that there is the correlation isn't quite as strong as you're showing here, right? Especially with HSV1. With HSV2, I think it's a little bit different. If you get the infection, you're probably more likely to be established latency. With HSV1, not so much. So look into those statistics a little bit more, and before you move on with your experiments. And again, if you want to really back up some of these correlations that you made, the best way to do it is numbers, right? I mean, you put some numbers in there that show, look, we've got these numbers that show there's a real strong statistical correlation between X and Y, right? Those really are not in the document, so I again infer that it's probably just not all that strong. Some of the techniques you want to be a little bit clearer on, things like PCR PCR is polymerase chain reaction. It detects changes, or it detects the presence or changes potentially in nucleic acids, right? It does not detect amino acids at all. So you can't detect the presence of a protein using PCR. You can detect the presence of a nucleic acid, and a very specific nucleic acid, if you use the right primer sequence. You made some mistakes like that throughout on your descriptions of things, so you want to instill enthusiasm in your reviewers and in the people who you might want to get your money from, and misdefining PCR and things like that doesn't really instill that enthusiasm in that sense, right? But that, believe me, I know you guys are really moving in the right direction, but try not to make those kind of mistakes. I think what seems to have happened here is, I think there are probably several of you who know what PCR is exactly, but maybe you didn't all read over the document really carefully and catch those errors, right, and things. So I think it's important that we have a team of people, you all look it over and catch the other people's mistakes. We all make mistakes. I make plenty of them, and some of them are really stupid, right? I mean, I'll write a paper and say something incredibly stupid in it that no trained expert virologist that's been as old as I am should ever make, but I make it, right? And so I depend on my teammates and the people in my lab to say, you know, that's not really true. I don't know where you got that from, but you must have dreamed it last night or something, right? So, yeah, check these kind of things over carefully, and I've pointed out a few of them that are in here. The last thing I want to say about the whole M23 thing, I think, as we've talked about, you could detect differences in the tetramer structure, perhaps, maybe because you got less of one subunit and more of another. Moving on, I doubt there'll be any mutations, but even more important than that, you have to be really careful when you're even looking for downregulation of a particular protein during a viral infection. The reason being that viruses like herpes simplex virus and a lot of other ones, they downregulate a whole bunch of proteins in cells. And so I would be willing to bet that if you took 10 host proteins and measured their levels during herpes simplex virus infection, 5 of them would be down considerably lower than they were before the infection. That's just because herpes simplex virus is a global inhibitor of host protein synthesis, not at the same level as some other viruses. So even if you find that M23 is significantly downregulated, you have to defend that in a way that says this is a little bit more specific than the fact, because there are lots of proteins that are going to be downregulated by a herpes infection. For example, if you found only one subunit was downregulated and not the other so much, that would be, I think, a really interesting finding.
So what you could do there is you could compare it with GFAP, which is a intermediary filament protein present in astrocytes, or with beta-actin, okay, which is a microtubule. And so if you do a ratio between that and your M23, you will know whether there's a differential regulation of it or not.
Even if it's not differential, it doesn't mean it's not important, right? I mean, it can still be very important to the glycoporine system that you're talking about, right? But it's not necessarily something that I wouldn't expect to find. If somebody said, I did this experiment and look, this protein was downregulated when I infected the cells with HSV, I would say, yeah, there are hundreds of proteins like that in the cell, right, that are downregulated by HSV infection. So that's kind of what one might expect. So, is that?
Yeah, so the other thing, so will the virus kill the cell, or will it just survive in the cell, do you know? On the literature?
I think survive, yeah.
Survive.
It should survive.
So you
long
should look
enough
for cytopathic effects in your culture. Because
Yeah.
if it kills the darn thing, then you've got nothing to measure.
It
right?
kills epithelial cells,
Right?
for sure.
Yeah.
But neuronal cells, at least the ones that it establishes latency in it, doesn't kill. However, neuronal cell
An acute
models
infection
may.
may actually kill them.
Right, it may kill these, again, I would, the other factor here is you're very careful about not using animals, but you don't make it clear that you're really not using animals, right? I mean, so these cells that are gonna be from the brains of mice, who's gonna take them from the brains of the mice?
We will be ordering them
Okay.
separately.
Right, you wanna make that really clear that they are from the brains of mice, but we're not involved in removing them from the brains of the mice, right? They're gonna come.
Commercially available.
But I would actually suggest just what was previously said, that I put in here, why are you using a rat model here? Again, it would seem like it would be maybe even easier to just get human astrocytes. Do you have like immortalized lines, or do you have like?
We got primary cells and we got immortalized, we can give you all. So
And
it's
so
not a
it
problem.
may even be easier and more reasonable to use a human model, right?
Yeah.
Initially, you said you use the red because they want to propose red, want red, you explain
Yeah.
that.
Originally,
And then you told them it's impossible for 1 year to finish red.
Right.
So they changed to red cells, that's the reason. to use
Yeah,
red as
so
precise.
the problem with doing animal studies as an undergrad is not all that easy, right? You've got to have an approval through IACU, how you're gonna, the animals, then you gotta learn how to handle animals. I think it would be very ambitious to do it as an undergrad. My guess is if you could accomplish what you're doing in vitro, that would itself be huge, you know?
Yeah, because they have lots of class work.
Yeah.
That's the problem, not like
I
graduate
think that's a
school.
good idea. I mean, you could, there's always a possibility of trying both lines,
I
right?
can-
and see-
yeah, I
But I
can-
would try the human line first, probably. if I was you.
Yeah, and the thing is, it's gonna take you a while to learn how to do tissue culture. You're gonna end up contaminating your cells. You're gonna, learn how to count cells, how to photograph these cells, how to immunostain these cells, how to infect them with the right dosage. It's gonna take a bit to figure out how to culture your virus, how to grow that up, how to titrate it, So there are a lot of techniques involved in just being able to get that experiment to work properly, and then be able to reproduce it. You gotta do it, several times and see you come up with the same result or not. So that's gonna take a while for you. And then you've got other techniques in there. You got protein extraction, you got Western blots, you have a lot of
I would say
techniques.
just like, I actually thought AIM-1 in and of itself would be a big accomplishment, to be honest, right? I mean, so that means that you've done all the things that you said that you have to do, right? Learn how to start, and look, we contaminate ourselves in the lab all the time. I've been running the lab for 23 years. I throw out cells every month, right, that have been loaded with bacteria and things, so it's not trivial. Just to get through AIM-1, do a fax measurement or something of that sort would be a big accomplishment, I think, right? And I think that should be a big goal. This is a little bit overambitious. It's fine, though. It's better to be, I think, overambitious than underambitious, right? But keep in mind that having to learn all these techniques, get a fax to work, actually get a real quantitation of this, right, would actually be a big accomplishment, I think. The other thing, of course, is your budget in here is way lower than the budget you set up there. And this budget is, that budget's somewhat more realistic, I would say. This one is.
Yeah.
I think,
Last night I told them.
I think what may have happened in the grand scheme of things, we may have missed a zero in there somewhere.
Yeah.
How in the world we got that low?
Well, I think, it's kind of like you added one kid of this, one kid of that, and,
Yeah.
that's just the starting point to get things to work, right? I mean,
Right.
Count like the number of years and times.
Yeah, so you probably need like
Okay.
throw out cells.
Yeah, sure, move on.
Jane, you have some question you want to ask?
Yeah, for the flu, the part, I have some question for you. You said in A1,
Mhm.
you try to use that method to detect the amino acid sequence. A1, you detect amino acid sequence? Why?
The
Looks like
amino,
you're a little confused there.
the amino,
Go back and flip back.
Yeah.
oh wait, can we go back to AIM-1?
Yeah,
Yeah.
pull up whatever slide you need.
Yeah.
Yes, sir.
It's usually me.
Oh, the TV probably turned off.
Oh, did it shut down all the way?
Yeah, I think the TV turned off just 'cause it's been a while. Yep.
Okay, it'll take a minute to boot back up.
Was it approached anymore?
Yes.
Okay, please. Turns on.
Here it comes.
There we go.
There you go.
Okay. Sorry.
Could you repeat your question, Dr. Zay?
You said the A1, you try to
Yes.
analyze the amino acid sequence. What's the point? Why?
Oh, the point of analyzing the amino acid sequence would be to essentially analyze whether there would be a change. But now we know that
A1 basically you're trying to see the expression level change.
the expression level change. Yes, but we want,
Do you want me to go back one?
this is A1.
This is.
I know, but like, okay, never mind.
Okay, but then we wanted to also analyze the RNA level as well as the M23 level.
Yeah, that's the level change, not the amino acid sequence change.
Okay.
Alright? And also you write there, Western blotting isolate specific amino acid sequences.
With,
Yeah, just as Stephen already pointed out, some of those terminologies are not very clear.
not very clear, being more
Yeah.
clear about them.
Yeah, Western blotting detect the protein, the size of the proteins.
Mm,
You cannot analyze the sequence.
okay.
You just interpret that as the use of very specific terminology, you know, which among casual conversation may be finally adequate, but when you're getting to very clearly defined and specifically defined techniques, you make sure the terminology is correct.
Okay.
Right, yeah, concept, right?
Yep.
For the M23, you say 40 kD of protein,
Yes.
that's run in a native gel.
Yes, on, this is aim two with the,
Yeah.
This one or the slide before?
Yeah.
this one.
Okay.
So with like, okay, so in one of the literature reviews that I read with rat astrocytes, when the protein sequences run on the gel, the M23 and M1 tetramer are both found in the 40 kilodalton range of the gel,
Native gel.
of the native gel, yes.
Okay. Do you expect any protein interaction there in the native gel?
With the protein interaction, no, they're both isolated into separate strands. The M1 is found,
So specifically seeing that the tetramer will be disintegrated into monomers in the native gel.
yes, in the native gel, they will each be separated into monomers. So in the 3.2%, you will find the M1 tetramer, and in the 4% agarose gel, you will find the M23 tetramer. They'll be separated by the concentration of gels, because the M23 weighs more in size as opposed to the M1.
Okay. So the 40 kD you are saying in the native gel.
The 40 kD, no, in the blue native gel electrophoresis.
Okay, in the native gel you can tell the size of the protein.
Yes, in the native gel, you can tell the size of the protein.
So in the denatured gel, what's the difference between nature gel and denatured gel? Native and denatured gel.
So in a native gel, you can tell the, well, in a native gel, you can essentially tell the overall size of the protein, but as opposed to the denatured gel, this tells you the separated bands of the protein as it runs on the gel.
So the denatured gel tells you the size, but not the native gel. Native gel can't tell you the size.
The blue native gel can, yes it can,
You can?
yes.
So there's no difference from denatured gel. There must be a difference there.
I think what's happening is on the blue native gel, you're getting you're getting complexes to separate from each other on the blue native gel. There's no real ability to tell the size of something on a native
sure.
gel. But what you're getting is a separation of different complexes. It's actually shown on the picture there. You see those big complexes
Yeah.
that are separated on the gel? And then you run those complexes into a denaturing gel and separate the bands in the complex based on size.
Okay,
So the first gel is separating based on just the structure and gross complex size, but you can't tell the size from it directly. But it's
Okay.
something about a structure. It could be, you know, it's shaped a certain way, it could be it's just huge, and one's round, one's a little bit square or whatever, right?
Okay.
So those separate in this BN, in the blue native gel, in a certain way. And then you run those complexes into an SDS page gel, which pulls them apart and separates them by their size. So it's the SDS page that actually does the size separation.
So that's the concept issues, right?
okay.
Yes.
Any more?
No more.
Okay. Let's go back to a little bit more general question I have. You had three major aims.
Yes,
I understand that aim 3 is totally dependent on success of the aims as
Well,
well.
was, yes.
Yeah.
I got that, okay. Is aim 2 totally dependent on the success of aim 1?
aim to determine if there's a structural change.
Aim-
Not exact, no, aim two is not totally dependent on aim one, because in aim one, we just wanna analyze if there's a, change in the amount of M23 produced or reduced in aim one, as opposed to aim two, where we're trying to determine if there is a protein folding change.
Aim one is
within
kind of like
M23.
setting it up, kind of,
Okay.
and then aim two would be like actual.
So can aim 1 and aim 2 be run at the same time, or they need to be sequentially run?
Aim one and aim two can be run at two different times.
Or at the same time?
Yes.
Okay,
Sorry.
yeah, okay, good. That's just giving me, so your timeline might work out.
Yeah.
Okay,
Yeah.
because these all not being a neurobiologist, but having read a lot of proposals and projects of similar, it's gonna take 200 times longer than you think to do M1. And Stefano said that M1 is a challenge in itself and a success story if you pull it off. So don't wait. If you can start M2 in parallel with M1, don't wait to do that, because you may end up finding out M1's taking a whole heck of a lot of time. Then M3, I understand, the modeling part is dependent on whether you have something to model or not.
Yes.
Yeah.
Okay, I got
If
it.
there's any change or
Right.
anything that we can detect.
Okay. Good, you gotta keep in mind that these things will take you a lot longer than you think. Back to your funding, I'm really glad to see the 10,000, I know the 1,400 wasn't gonna work.
Yeah.
I don't have $10,000 to give you. You alluded that you were gonna be very successful in the next few months of finding $10,000. How are you gonna do that?
Through a combination of grants, we're gonna
I need
set
more
up.
specific answer than that.
Foundations of
Yeah,
the
for
Alzheimer's
funding.
Society of America, they have a grant proposal due in the middle of March that we intend to apply to, and also the Alzheimer's Society of America, which those are two grants we can possibly apply to, but that is toward the beginning of April for the
Okay.
second deadline.
For the first one, if you need to have a proposal in their office by the middle of March, when do you need to have that written and approved and sent through the university approval process?
A month before it's due.
And what's today?
February 19th.
That proposal better be worked on this weekend if you're trying to get it out in time. Because you need to write it, your advisor has to review it, edit it, and then run through Aura and the whole process. Okay, so, yeah, middle of March is now.
So tell me something, I mean, how do you expect the Gemstone project to work if you don't have any internal funding at all?
We have some internal funding. We
What-
encourage them to
yeah.
pursue
Other resources.
other resources.
So what kind of funding can you provide for a project?
We provide them a baseline funding to get started of $600, okay, which isn't much. Frequently, the mentors find resources available, maybe not money, but supplies. And then we encourage them to look for outside funding, and if that still comes up short, then we have supplemental funding upon request, but they have to show a real need for it. The fact that I missed application deadlines for external grants is not a good explanation for asking for extra
There are no
money.
internal granting mechanisms that they can apply for, University of Maryland doesn't.
There are some for specific types of research, but these types, there's none internally unknown.
Yeah, and my gut feeling is it's not gonna be competitive with the Alzheimer's Society.
Do you
Okay.
have
No.
any perhaps ideas on other grants or places we should try to aim for them?
I mean, I really don't know.
Because I know I've been trying to talk to Alana about like applying to different places. She said she was busy with like her med school applications, but then after that she'd be like
Mhm.
happy to help me. Because
Have you looked into the launch UMD process?
I
Yes.
was actually just about to say that. So
Okay.
I'm currently doing the Launch UMD project for a different organization.
Mhm.
So now I'm pretty familiar with that
Excellent.
process. I know how to make a good application. So I'm very confident that we can get one.
Right. Launches are our internal crowdfunding, and we have had many teams very successful in about the $5,000 range
Yeah.
of,
That might be a good way. The other way, of course, is that, you just try to get as much stuff free as you can.
Ask
So,
around.
he's got some free cells for you. I mean, and a variety of, there's lots of this stuff that, it does cost money, lots of the stuff you can get free. To be honest with you, a lot of these projects end up getting funding from the lab that they're in, and so that has to be, that's a problem, right, in some ways, right? I mean, that, but you gotta keep you gotta be very cost effective when you're doing these, so you don't wanna do really expensive stuff. Cell culture, I don't think is hugely expensive, right? So I mean, that's, Dr. Zhang has virus in his lab and things like that, right? So those things are not massively expensive undertakings, right? I mean, I don't, what would be expensive here might be the flow cytometry and other things, right? I mean,
Antibodies.
Antibodies, right, yeah.
Those things cost,
Yeah, those are expensive. Antibodies, I mean, one antibody is, in the $500 range, right?
Okay, so the thing is, there are sources for getting very cheap or free antibodies, okay? So they're set up by NIH and some other institutions whereby you can get them for $10, $20, $30 at the most. So basically you're paying for shipping costs. And especially the more, I mean, if you have a very special antibody, it's a different, but there are places you can get them. I don't remember them off the top of my head right now, but if you send me an email, I can try to dig them out for you.
Thank you.
Good resource.
And that's one thing, I work with HIV, so they have the AIDS reagent program from NIH, where you can get lots and lots of stuff for it, right? But I mean, unfortunately for HSV, it's not quite as organized, right, and it's a little more difficult. But you're not looking, the antibody you'd be looking for would be to the host, the M23 protein anyway.
mTOR, you need GFAP, you need beta-actin,
Right,
you need
that's
some of these, so those things we could probably get close to nothing. Yeah.
Okay. Do you have follow-up technical questions you'd like to ask?
I think that's pretty good.
I have one more general team type question. You're getting launched on a very ambitious path here. I think you brought yourself to a very good starting point. The reaction I've heard from the experts in the room is encouraging. Okay, so I would encourage you to go forward. Keep your ultimate goal within reach. Okay, but it's a long road between now and your senior thesis that you so gallantly said will be just written. Okay, so you have 6 members of your team. You all have 6 different lives. Okay, how are you going to hold each other accountable for making sure that everyone's pitching in, everyone's doing their share, everyone's getting done what they need to get done? What's your process for making sure that all 6 of you are viable and invested members of this team?
Well, actually, Robin just
Yeah.
recently.
Okay, so originally we just kind of, when we have our team meetings, we're like, all right, here's what we need to get done, here's what we're gonna do. Maybe sometimes we split up or we partner up and divide the work like that. We also have a Google Calendar that we can all see, it's like all synced up to our Gmail accounts. In that we put in like all of our set deadlines, we had like this in our calendar, so we said, all right, we have like this tonight and we need everything done, by this date, we want this done by that date, so we set deadlines with that. And then so we can like assign work based on that too, but I know that things come up, and maybe like someone gets really busy one day, like I know last Friday for me was super chaotic and busy, and so like I didn't have enough time, to maybe dedicate what I should have, so then in that case, it's kind of like shoot each other a message like, hey, I did as much as I could, can somebody like look at this between classes or something, and someone else is like, yeah, sure, like I have my computer, I'll go look at it, so we do that, and then if we see that people are slacking, we kind of just, bring it together and say, look guys, like we all know, we all have things going on, we maybe don't have like as much time as we'd like to dedicate to this, but we are a team, we're 6 people, like we can do this, we're awesome. And you can also, in the Google stuff we use, Google Drive for all our files, you can also like see the revision history too, so you can like see who is or like is not doing work too. But we never ever call somebody out on it, we're just kind of like as a team, like hey, we all need to like work together better, we all need to be accountable for our work.
And there may be a point down the line where you need to call somebody out. And that's part of being a teammate.
So who's gonna teach you all these techniques? You have the resources to teach them?
We,
What's the
like,
option?
so actually, like, in our proposal and everything, like, I don't know if you read into the appendix, we actually have, like, the different steps to go through these different processes, but I also do research on, like, campus in an extracrystallography lab, and I was also asking my PI whether he could, like, help us use some of these models, because we have an immunofluorescence staining model in our lab, and things of that nature, and he was just like, would gemstone by any chance, like, happen to find me? I was like, no, but that would be something that,
Good
like,
answer.
huh? I was like, no, but that would, like, something, like, that would possibly be something our team could pay for. So, yeah, he could help us.
Teach
Do
you
a second.
university.
Yeah, I have a class to teach in 5 minutes,
Okay.
unfortunately.
Go ahead and if you would just turn off the recorder.
Okay, so I just wanted to say good job, you guys, and I can definitely help you follow up